From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 91, recorded on June 21st, 2023. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent, great to be with you from LD Lab Studio. And we're back on air, live streaming on YouTube.
Did we miss a month?
No, I think, it might have been an extra week or two just in the monthly churn with, I think you've been on the summer science tour, if I've got that right.
Oh, yeah.
And so I caught you just on a little intermission between meetings. I guess you were just at the ASM meeting and heading off to ASV. What are these?
Yeah, American Society for Microbiology last week, and now American Society for Virology coming up Friday, which is in Athens, Georgia. And before ASM, I was in Philadelphia for a one-day symposium. Before that, I think I was at the aquatic. No, I was somewhere else before that. I was at the, Philadelphia. Before that, I was in US AMRID with Rich Condit.
Oh, cool.
And then before that, I was at the aquatic virus meeting. I did a symposium in Washington. In between those, before that, I was in Montreux. And before that, I ended my class, my virology class. So actually, after ASV, then I have a little break until August. And then in the fall, the travel starts again. But we'll going all over the place. I'm going to Milan, Italy. I'm going to Hong Kong. I am going to Germany for the giant virus meeting.
Oh, yeah. Great
lineup. Been to Hong Kong? Ever been to Hong Kong?
I haven't been to Hong Kong. Let's see. The closest might be Singapore and in Thailand. But yeah, I'm kind of on summer hiatus from travel right now, and we'll pick up again later this summer in August and then into the academic season. But yeah, good to kind of catch the breath here and take a little break from travel. But
well, I want to do some work here at the incubator. I have an assistant now. Who is helping me do stuff, and so I want to take advantage of it and make some videos here in studio and so forth. Anyway, welcome everybody. Welcome to TWiEVO. I want to thank our moderators before we start here. We have Barb Mack, we have Tom, we have Andrew, we have Steph. I think that does it for today. I joked at the beginning that we were going to have more moderators than participants here, but that's not the case. We have a good crowd of 30 plus. Welcome back, everyone.
Welcome.
Everyone's here to see your beard, Nels. How's it going?
It's a tough but fair question. Going fine. I'd say a few more gray hairs showing up from time to time, but running pretty well. Actually, it's been a while. I usually try to, on kind of an annual basis, do a clear cut, bring it down to a clean shaven scenario, kind of like when you try to restore a forest ecosystem, somehow like burning it down and then letting it recover, I think is part of the equation somehow. But it's been a while, so I'm probably overdue. Maybe that could be a summer hobby right there. So stay tuned maybe for TWiEVO 92. Maybe I'll come in with a little different look.
Let's see where folks are from.
Yes.
Tonight or this afternoon. I know that Barb Mack is in the UK, right? Because, Barb Mack, UK. And then Nicola is in Italy, 26C. Actually, it's not very warm here in New York. It's quite mild. It's 22C here in New York. What do you have there in Salt Lake?
Yes, very similar. Unseasonably cool here. And by the way, happy midsummer, everyone. We're on the longest day of the year, I believe, in the Northern
21st.
Hemisphere. Yeah.
That's right.
But yeah, it's been a really nice, lush, kind of cool spring moving into summer.
By the way, Tom's here. I don't know. I guess he's in Madison, Wisconsin, or Oregon, one of the two. So he's mentioning Eugene Koonin,
Hmm.
So I ran into Eugene in Houston. He was at ASM. He got some
Oh, cool.
award.
Yeah.
And then the previous week, I saw him at a symposium for Koistya Chumakov, which was in Bethesda, and that I didn't realize. So Kunin actually trained in the Soviet Union with Koistya,
Wow. Yeah. Yep.
and the rumor has it that Kunin was so bad in the lab that Koistya said to him, "Go work on a computer," and they became a computational biologist.
It's all downhill from there. Yeah, I think he was just inducted into the Academy, American Academy of Microbiology. I was lucky enough to be a reviewer on his inaugural paper that I think is just out recently in
Is that
MBio.
right?
Yeah, it's a fun one. It has a little poxvirus angle to it. So, yeah.
Philip is in Wales, UK. Hello.
Hello, Philip.
I know Peter is in Boston or Cambridge. Yeah, I know this.
Yeah.
I'm getting used to all these people. Geraldine is in Michigan. I know that Andrew is in New Zealand.
Welcome.
You see,
Good.
folks. You see, I pay attention. I'm paying attention here. M.E. Wheeler is in Helsinki. A warm summer evening at 24C. I really like Helsinki. It's a fun.
I've never been, but on my list. Yeah. Welcome.
There's been a lot of discussion about Asgard and Norse gods, so we're going to get into that. That'll be fun.
That's right. We're going into the Norse mythology space here, in honor of Midsummer, where the sun's not setting at the moment, it's staying light.
Tom's in Madison, Wisconsin, which is where we were a year ago, Tom, roughly at ASV, and I met you there. That was very cool.
Oh, yeah. I was at that meeting, too. That was a great one. We did some fishing from Rob Kalita's dock, as usual.
So Pete is from North London, and so is Thomas. So what is it about North London, anyway? Marianne is from a boiling hot Norway.
Ooh, there we go.
Barb Mack is from the West Midlands. Toby is from Inverness, Scotland.
Welcome.
Peter S. is in Boulder, not too far from Nels, right?
Other side of the Rocky Mountains. Yeah, we're holding down the west side here.
Did a podcast last week with your colleague, Jason Shepard.
Oh, good. This Week in Neuro.
Yes,
Twin.
This Week in Neuroscience. Yes, but this is This Week in Evolution.
Yeah, so we've been hinting at it. Let's jump into it. So we're doing a paper today. This is an open access paper, inference and reconstruction of the Hedal archaeal ancestry of eukaryotes. And so this gets at one of the, before we get into the science itself, let's talk about the scientists here and lift up some of the people who did the work. So first author is Laura Emma, and the senior author here is Thais Etima. And so the work started in Uppsala University in Sweden. The lab has since moved to the Wageningen University in the Netherlands. And the first author, Laura Emma, is now running her own group, I think, at Orsay in Paris after a stint at the CNRS. And so, and actually, here an international cast. We're going to talk about some metagenomics. We're going to talk about some phylogenetics, some comparative analysis of genomes or protein sequences that is trying to get at one of the sort of fundamental questions of all of evolution, and also sort of speaks to what's been a long-running debate in evolution over whether the tree of life should be organized into two domains, which was sort of an old view, three domains, which was a view that was also that sort of arose the Carl Woese days, in bringing in some of the first molecular characters in phylogenetic tree building. And then we've returned in the last decade or so, maybe 2 decades even, to back to a two-domain view of life. And what this has done is it moved our ancestors, the eukaryotes, into the clade or the grouping of archaeal species. And based on some, in particular, more recently, about 10 years ago, some sampling of archaea, these Asgard so-called archaea from deep hydrothermal vents that then were named, as we'll kind of be stepping through in some of these cases, some of the Norse gods of mythology, but also now, in this paper and in some other recent work, in some cases semi-competing with this work, metagenomics and the comparative approaches, using the increasingly powerful tools of genomics, is really opening up an entire sort of new world of diversity among the archaea based on those discoveries from about 8 or 10 years ago, and really adding, I think, a more rich picture, a more nuanced picture, a robust picture of the relationships on the tree of life. And so the reason, so what's the big deal here? If it's a 2-domain or 3-domain branch of life, I think what matters here is to really get at sort of these fundamental questions, sort of curiosity questions that I think everyone has, whether you're a sort of formal card-carrying evolutionary biologist or not, which is, where did life come from? How did sort of the complexity of life happen? And so the key groups here, among those either 2 or 3 domains of life, are the bacteria, the archaea, and the eukaryotes. So you and I, Vincent, we're eukaryotes, for better and for worse.
Yeah. Although I'd like to be in Asgard. I don't know.
Yeah, pretty cool. I agree. Although sort of ripped from the headlines, and unfortunately in sort of a potentially tragic way, deep sea exploration has been in the headlines here the last few days, and some of the challenges there. But so I think a lot of the sampling here is actually from these deep sea events, but with robots, with drones, underwater sampling collection. So bringing up these samples, these new species, newly described species of archaea, and then getting not only sort of the cells together at the beginning, sequencing some of the first genomes, but then going out and now sampling around the world and doing metagenomics, which is just sort of, you've got a complex collection of critters, in this case microbes, from soil samples, water samples, mud samples, etc. And then you just sequence all the nucleic acids you can get your hands on. And then in the computational steps, try to put this all together. How do the genomes sort of arrange from species to species? And can you then, as you doing this sort of some of these newer, more powerful approaches, trying to take that massive collection of messy data and actually get entire genomes of different species. And so this is at play here in what they're calling MAGs. These are metagenomic assembled genomes, which are starting to cast light on this question.
So Nels, let me ask you. So it was Carl Woese that first proposed the 3 domains based on ribosomal RNA sequencing, and I wonder what he would think here. But he didn't have access to all of these genomes, so he might say, "Yeah, this makes sense to nestle the eukaryotes in with the archaea," right?
I think he would. And maybe one of the, in Champaign-Urbana, there's a great lab, Rachel Whitaker's lab, that works on archaea, works on the bacteria and viruses of archaea as well, kind of following in the footsteps of Carl Woese. And, I think she is also starting very much in this space of let's look where the genomic data goes. And in this case, the 2 domains. And in fact, here, I think it will, they'll even be, as I think the words that you used are there, where do the eukaryotes nestle into the tree or among the archaea? But let's not forget the bacteria for a moment here. So this is kind of pushed to the side, but still a key event, obviously, is the ancient endosymbiosis event, the origins of mitochondria. And so this sort of also is an important part of thinking about where did eukaryotes come from? What's our last common ancestor with the archaea in this case, which we'll kind of dig into a little bit more. But what are the sort of genetic contributions that then lead to the cellular processes? And I think that's one of the kind of exciting things here that we'll talk about, is to not only do that sort of genomics approach, the comparative approach, and we'll be talking a lot about trees and how they arrange and some of the details there. But I think underlying that and starting to emerge from this work and in the field, which is why it's worth it to spend some time together talking about this, is to actually start thinking about what does that mean for how a cell works, the cellular processes that were there sort of in those last common ancestors that are inferred to be roughly 2 billion years ago. And how does that then influence life all the way today and the diversity of life around us? How are we related to each other? And how is evolution sort of unfolding in ancient times, but in actual time today, right before our eyes, with some of these microbial populations, right, that can replicate themselves in such a short time span that we can see them evolving in real time.
And just to be clear, the first to evolve on Earth were the bacteria, correct?
That's right. Yeah, exactly. And then sort of the Carl Woese tree shows this branch point. So you have the bacteria on one side, the archaea coming out, and then a sort of a long branch at the bottom with the eukaryotes out to the side. My hands are going off the screen here, but and so what's happened then in the last few years is that branch point for eukaryotes has been changed. It's been moved around. So in either closer next to the archaea, or in this case, among them. And that's sort of been pivoting as more comparative analysis has been added. And so there are two.
Just to interject, so they
please.
note in this paper the ribosomal RNA, which he used, are prone to artifacts, and that's probably why that branch is being moved in, right?
Yeah. So there's a couple things at play here. So one is just the number of species, the samples. Right. And so, a phylogenetic tree, a tree of life, is a hypothesis. It's not sort of a set in stone thing. And so the more samples that you get, the more that's going to change not only how many sort of branches there are in the tree, but in some cases, the arrangements where the branches come out of the trunk, that starts to move. So that's kind of factor one, is as we're sort of getting a hold of more of the life around us. Factor two are the techniques you use. So, and maybe that sort of comes out of your point, Vincent, which is if you're using ribosomal proteins, it's like one snapshot of all of the potential characters that you could use to compare. And so, remember
RNAs, right?
Yeah, ribosomal RNAs, or in a lot of the work too, the genes that encode the ribosomal proteins and collections of them. It's kind of, you might think that, well, now that we're in sort of the genome era or post-genome era, why don't you just take the whole genome and compare those across species? That turns out to be really tricky for several reasons, and kind of counterintuitively doesn't necessarily give you a better sense of how two species are related to each other. And in fact, the biggest problem is there's more than, you can't just, like, genomes are sort of living, breathing genetic documents in a sense, right? And so you're adding genes, you're taking them away, you're recombining, you're swapping information in different, settings. And so to do it, the more that you try to compare, the harder it is. And so the compromises that have been made to get these really broad swaths of diversity from bacteria, archaea to eukaryotes, is to pick, like you're saying, a key set of ribosomal RNA sequences, or the DNA sequences encoding the ribosomal proteins, or in this case, they'll propose a different set of genes that go outside the ribosomal ones, and we'll talk maybe a little bit more about that. And then finally, actually, the technology is also advancing, not only sort of in the genomics, so the sampling, the number of samples you get, et cetera, and not just in the sort of sequences that you choose to compare, but then the models that you use to sort of try to get a better sense, so you're not fooled by some of the patterns of mutational processes that might fool you, might produce artifacts. Basically, there are these sort of patterns. Long branch attraction is one, where two seemingly close related species based on a tree are actually much less related to each other. It's just the way the model was set up, or the way the data was analyzed, has brought them together in a way on the tree that doesn't represent the real life descent of the ancestors of those two species, or collection of species.
Let me pick up a question here from Marianne before we move on.
Fantastic.
Do we have common ancestors, or did you say the mitochondria comes from the eukaryotes and we got them from them?
Yeah. So that actually, thank you for the question. It actually nicely crystallizes one of the main points of the paper too, which is to get at that, what did the last common ancestor look like? So if we put this eukaryotic ancestor together with the archaeal ancestor, that's sort of the question this builds from. On the mitochondria side, so that's sort of a different biological event, where the notion is that our ancestor eukaryotes existed without mitochondria. Mm-hmm. And then there was this endosymbiosis event, where basically that ancestor, or population of ancestors, engulfed a bacteria. And in most circumstances, you would imagine, and this happens every day right before our eyes, is if you engulf a bacteria, you usually digest it, or the bacteria infects you for a while, your immune system knocks it out, and then you sort of move on. But in this case, it became an alliance in these ancestral single cells where not only was the bacteria engulfed, but in this case, it persisted and then has evolved. It's degraded, so it no longer is you can't replicate on its own. It's been subsumed by the eukaryotic cell, and then the mitochondria obviously has become a core part of our own energy production, of our own metabolism. And so this has been, probably one of the most long-running and successful symbioses that our species has ever observed, and that our ancestors participated in.
That we're not sure where that occurred at the level of a archaea or a eukaryote, right?
Oh, so that was later. So that was so the notion here, correct, is that the archaea and the eukaryotes split, and then in the eukaryotic lineage, the mitochondria was engulfed, or the bacteria that became the mitochondria was engulfed. There's other endosymbiotic events, similar ones that have led to chloroplasts, have led to other organelles, basically, that are sort of functionally equivalent or related to mitochondrial biology. Yeah,
but we don't have any eukaryotes without mitochondria. Right?
Correct. That's right. So that was a very early event in the descent of the eukaryotes. That's exactly right. Okay. Yep. Okay. Yep. Great question.
So Andrew wants to know if Ran is the goddess of RNA. Of course.
Okay. So here we go. So as we kind of hinted at, so a lot of these first species were discovered about 10 years ago. And so here there's a discovery of new species, but based on that sort of breakthrough discovery of being able to recover, actually observe some of these cells, sequence the genomes. And now, so the sampling is going in a much less sort of intensive view. It's just sampling stuff and then sequencing it. And so here the authors gain or propose that they've, through just sampling, and I think there's like 10 or 11 different locations around the world, Mexico, China, some Scandinavian locations, and get 60 more species. And then, put this into their sort of.
And Nels, when they do this, they're going in places that they're likely to find archaea, right? They're going to aquatic sediments and hydrothermal deposits, right?
Yeah, that's exactly right. Yeah, and that's an important point, because you probably can't go to your backyard soil and find relatives. Although I never be sure. I mean, you never know how far these can go.
You might, if you have a cow in your backyard, you might.
Absolutely. And there's also, what is an extreme environment? So, and the authors get into this actually. So, the idea that they're putting out there is that some of the original archaea were living near these in or around these underwater hydrothermic vents, et cetera. And it was but a subset of them, which is in the title of the paper here, the Hedal archaea. I think that's the son of Odin, perhaps, in Hedal in Norse mythology. Those actually live in more mesophilic or moderate temperature environments. Mm-hmm. And so that might also be sort of an important kind of stepping stone in the emergence of eukaryotes from in and around that group, or that association with that group. It might hint at some of the steps that happen in the deep history leading to our ancestors. Okay,
so now over where you are in the Salt Lake, there are probably some archaea there, right?
Yeah, it's wild.
Yeah.
Absolutely right. Yeah, and undersampled, actually. I think we could do a field trip here.
And Nels has them in his backyard.
True. Then we just had some kind of a dust event, so they probably blew over, and there's probably some hiding in my beard right now. Maybe after the podcast, I'll just go and try to sample some onto a petri dish. Okay, so 60 more species adding to the tree. And so that's sort of and not only adding to the tree, but as you said, Vincent, so it's not just the traditional ribosomal sequences here. They're adding to, or they're using a sort of a different set. I think they actually start with some of the ribosomal, that's sort of the traditional, and there's a set of, what's the number? 56, I believe, genes that they concatenate. They basically put head to toe all of those sequences, line them up among all of the samples, and then use that information. The software program, the pipeline, uses maximum likelihood to sort of make a tree of probabilities. What does it think is the most likely arrangement in terms of the relatedness of these? And so, by the way, when you look at these trees of life, these phylogenetic trees, the branches, there's usually a number beneath it. So there's a little like legend that says like 0.2 substitute, and that what that represents is the substitutions per site. So there's information depending on how the tree is drawn. And in figure 1, actually, the tree is drawn this way.
Have a look at that.
Yeah. And so there you can see that key at the bottom, or that that sort of scale bar, that 0.2, that substitutions per site. And so that gives you a kind of a calibration of how these 56 sequences are related to each other. So if that length between the branches is really short, there's very few substitutions per site. And this, a site is a amino acid in this case. So the proteins, the 56 proteins, are strings of amino acids. And that length on a tree is equivalent of, at every site, having on average 1/5 of a substitution, if that makes sense. So obviously you can't have, like, it's either a substitution or not for any individual case, but as you average that over the whole distance of the tree, that's how you get to that number. And so there's a lot of diversity here. And this, on the left side, those sort of green shaded area of the tree, those are the Loki archaea. These were some of the ones that were discovered, 8 or 10 years ago, a big splash in the field of evolutionary biology. And then as you go sort of across the tree, here's where the branches are being filled in by these metagenomic approaches. And so even in the last 10 years, having that first sort of set, or that original one, immediately, you can, the power of comparative genomics is pretty clear here, where coupled with more and more sequencing, where now you can just start to put together all of these, or infer all of these new species out there. In a lot of cases, no one's ever cultured these things, or no one's ever seen them alive. It's all based on the sequences, but that sequencing is getting so good that these are, not perfect, but they're pretty good approximations of entire genomes based on this.
So this is why we have the Asgard, right? We have all these gods and so forth.
Yeah, exactly. And the list gets longer and longer. Again, an international sort of group here. I think, mostly in Norse mythology, but there's some other names that are coming in some of the branches as well, and from some rival groups. Just kind of fun to see that kind of science unfolding here across the globe as people are sampling from their own kind of neighborhoods and geographies, and seeing all the relationship between, in these cases, the microbial populations and the archaea in particular.
What do you think, Nels? How saturated is this, do you think?
That's the thing, this is the exciting thing about microbes, right? Is that we're not even close. It's sort of like, the analogy to astronomy. How many stars are there in the sky? And as your telescopes get more powerful, like, that number gets bigger and bigger. And so I think we're just seeing, the tip of the iceberg, so to speak, in terms of the amount of microbial diversity. Similar thing happening, of course, in the virosphere, and some massive advantages or advances, thinking about giant virus genomes in particular. But that group gets bigger and bigger. And certainly, like, I think what we have begun to do is that we have representatives from sort of a lot of the major branches. And so it becomes harder and harder to discover something totally new or novel. Although here in this paper, they sort of, with this analysis, they add a new, clade, or a new collection of archaea in the Asgard sort of groups. And so, all, like, even sort of fundamental level discoveries in terms of opening up entire sort of wings of life that that we're just glimpsing at for the first time. Okay, so they point out that, so in the meantime, there's been other groups publishing as well. And I think another group maybe a couple of years ago did similar approaches using the ribosomal proteins and a different set of samples, and put the eukaryotes a little bit farther away from sort of that, less nestled in with the archaea, a little bit closer to those 3 domain of life maps. Still, I think you'd still consider it two domains, but maybe just not as close of a proposed alliance or origin story for the ancestor of eukaryotes. And so they make, a couple of points here. So one is, if you, as you add more species and more of that diversity, sort of redraws the borders, it puts the eukaryotic branches of life, clade of life, back closer. But they also point out, some potential problems or some upgrades to the nuts and bolts of the analysis here. And so one is to instead of using the ribosomal sequences, is they put together a new collection of 57 proteins, and these are all, so you can find them in archaea. So they're assumed to be of archaeal origins, but they're also found in eukaryotes. So that's important, right? There are genes that eukaryotes have that archaea and bacteria don't, and vice versa. And so you want ones that sort of cross between in order to really build these trees. Obviously, if you add back, if you try to get bacteria onto these trees, you can't use that same collection of 57 proteins because they're missing. Maybe not all, but maybe many are missing in bacteria. And that's important to think about too, as you're thinking about like the entire sort of tree of life. And here, the ones that they're proposing to use actually encode some of the core metabolic and cellular functions. And the reason or the motivation for doing this is that, kind of sprinkling across different functions will hopefully give a more true picture of the relationship. So the potential confounder of just using using ribosomal sequences is that the ribosome works as a single protein complex, RNA protein complex, in the process of protein translation, translating messenger RNA into protein. And so those proteins have co-evolved. And so they might, and even potentially independently between species that live in very different environments or shared environments, but are not as closely related to each other, the machinery of the ribosome might start to share some common features of coevolution because it's that same, it's basically doing the same job. And so their hope is that by kind of diversifying the portfolio of cellular functions, that some of that convergence or confounding might not be as bad. And so, and in fact, when they do the comparisons, it does change some of the arrangements of some of the clades in the tree. So, let's come back in a couple of years, 5 years, and someone might say, well, here's the problem with picking those 50 some odd proteins. Here's another artifact, and then propose another thing. And that kind of gives you a sense of the field, or, it's a scientific process. It keeps moving forward. And I don't know, in some ways, like, I think that's really, for me at least, intuitively, it's hard to remember that because when I think about evolution, or like, when you think about the relationship of us to other species, it feels like that is sort of set in stone. There is sort of a ground truth there. But as we're taking these modern species of today and trying to work backwards to infer the relationship as far as 2 billion years ago, there's a lot of sort of sand in the gears of really seeing what the truth is clearly. And so this is like any other scientific process. It's trying to get closer and closer to the truth as more information and more techniques and sort of information knowledge comes online. Okay, so that's part of the upgrade here. Another one is they try to take out the fastest evolving sites. So these are amino acids. If you compare, let's just take one of these 50 some odd proteins at one site or one position, one amino acid across, I think there's something like 350 comparisons here, total species, between 300 some odd archaeal species. There's 14 representatives from the eukaryotes. And so you take that like literally like 350 lines of letters lined up. And when the amino acid site has a lot of substitutions or is said to be rapidly evolving, they throw that one out. And the reason for this gets at the point I think you hinted at, Vincent, which is saturation. So the more changes you make over these long time periods, the chances that not only has there been one substitution that represents sort of the information that you have for your tree, but imagine if there's been 5 substitutions. And so you'd be underestimating how closely related any two species are. There'd be sort of this long branch attraction where very distant species would actually be more closely related, but you're missing those other 5 substitutions, become saturated. And so they just try to like eliminate that from their data, and that changes the arrangement of the tree a little bit. So that is a sense that there is some saturation here. And then finally, they do another sort of what's called SR4 recoding. This, by the way, the whole paper is like alphabet soup. There's so many acronyms here. It's just like, my goodness. Anyway, so try not to fall too far into down that slippery slope. But the recoding here represents that. So again, the degeneracy of the genetic code, the way you sort of try to discern the relationships or the mutational steps that make the sequences of the genes different between any of the two species or comparison points on the tree, make some assumptions about the exact kind of set of mutations that went into this. And so here they try to account for that by actually treating the differences in the amino acids that they see a little bit differently. It's sort of a conservative approach that tries to account for that. Again, sort of with the potential for the saturation of mutations in mind here, especially as you're comparing things that are so distantly related to each other, even among the archaea that are now being sort of sampled across the globe and have had billions of years to diverge from each other. These are very different from each other. The entire, eukaryotic clade falls within this massive set of diversity. And so doing that sort of brings us to the next figure, figure 2, which is, so figure 1 just showed all of the archaea in their sample, but now they have a second tree here that brings in the eukaryotes. And so, thank you for bringing this up, Vincent. So right in the middle there's a little gray triangle. So what they've done is kind of simplified the tree. Yeah, there it is with your cursor. That's the 14 eukaryotic representatives here. And so again, this is like they've just kind of compressed the single lines that you would see at the tips of the branches of the tree just into a triangle to make it a little bit easier to see. Actually, side note, like as someone who publishes phylogenetic trees, I really like these sort of simplifying ways of representing the figures. Otherwise, I just get lost in all of the, like even in that first figure, it's so detailed and there's so many species there, right? That it's just becomes like microscopic. And so it's really hard to, you're just sort of overwhelmed with the amount of information.
So basically they are replacing all these tips with just triangles, right?
Exactly. Yeah.
Like
it's sort of
see here. Yeah.
Correct. Yeah, exactly. In the major groups. And then you don't get quite as much info, but it's in some ways it's more digestible. And here, we're trying to look at the relationship between the major groups. And so if you trace that eukaryotic branch back, here's where you see it associated most closely with the Hedal archaea. And so that's sort of the big advance of this work that they put forward, is to actually make that robust connection, which puts the eukaryotes right in the middle of the diversity of all of these archaea. So very different from that Carl Wu's tree, the 3 domains of life, where eukaryotes were kind of hanging off on the edge there. And another important note here are those little circles. We see the black circles and the open circles. Those are meant to give you a sense of like, how robust is this relationship based on the data that they used. So this is so-called bootstrapping of the data. So basically what they do is take their data set, throw away chunks of it 100 times in different chunks, and then redraw the trees with some of the data missing to see how robust the relationships are. So if you have 57 proteins put together, you might take away 10 of them and then redraw the tree. Take away another 10, redraw the tree. Do that hundreds of times. And then the color of the dot, so if it's filled in 90 out of 100 times, the relationship along those branches recapitulates this pattern. The open circles, I think it's 70%, 7 out of, or actually it has the numbers there, the posterior probability, that's the arrangement, all the way from 0.7 up to 1. And so the darker the circle, the more confident they are that the relationship based on the data represents, or what they're trying to infer from real life or the ground state, the fact state, that they're capturing that based on the data they have in hand.
Nels, why is this eukaryotic branch so long?
Yeah, so that gets again at the the number of substitutions per site. So in here it's in that first one it was 0.2 substitutions. Here it's 0.1 substitution per site so it's actually roughly the same as above. It's just a smaller sort of interval there. And so what that reflects is that, I think in the divergence of eukaryotes from all of the archaea, there's sort of been these eukaryotes we're called eukaryotes for a reason, and we're not called archaea, in part because we've diverged. We've undergone 2 billion years of different evolution. And so that branch sort of represents probably the additional pressures on those gene sequences in the evolution to diverge from the sort of ancestral state or the last common ancestor.
Could also be undersampling for a long period of time, right?
Oh, absolutely. So, well, all of the sampling here is modern species, right? And so we don't have fossil records of the microbes. Yeah. And so, but it looks like that in its appearance. And it's not, it wouldn't be that there hasn't been, sort of populations of both archaea and eukaryotes over the entire stretch here. But sort of the evolutionary journeys of the genes have been very different as the species have diverged, the cells that house those genomes. And so, yeah, it's sort of a different character there. It's a good point. Okay. So, there it is, actually. I mean, it's a lot of work, years worth of work and sampling and all this genomics to sort of say, okay, we think this branch sort of sits here instead of like one little tick over. Kind of, maybe underwhelming in a sense from just like, okay, does it really so why does it matter that we put that branch there versus somewhere else? And so they start to get at that, actually, Vincent, with some of the, and this is the part of the paper that I think is in some ways even more exciting, more interesting, is to then start to think about complementary ways of thinking about the evolutionary paths of eukaryotes from archaea, and then to also say, can we use some of these cellular traits to actually understand our relationships even better? And so they move on to this sort of analysis using what they call ESPs, more of this alphabet soup here. So these are eukaryotic signature proteins, basically asking a roll call of which proteins are present in the genomes of some groups but not others. And so sort of presence and absence. And this gets at and that's missing from the trees. So in all of the tree data that we've looked at so far, in order for it to work, you have to have these guys all of these proteins on hand in order to make a full comparison. You might make some compromises. So if like some groups are missing some, you might just leave that out of all of the analysis or do sort of this hodgepodge analysis, but that can kind of really influence it. And so, part of their choosing the set of proteins they did is that they're highly represented across all of that diversity. Here, they're looking at this and asking, okay, what with other sets of proteins, these signature proteins, which ones are from eukaryotes, which ones are present and absent, or what's sort of the dynamics of these things in all of this of the sort of major groups. And this is shown in figure 3. It's sort of like a diagram of filled circles, open circles, which is just like a parts list of what you find in the color-coded groups, in some cases in others. And so if it's filled in, that's the presence of these so-called signature proteins of eukaryotes. And if it's not, it's either been lost or it was only a more recent acquisition back and forth. In this case, it's a lot of gene loss followed by duplications. So that sort of helps also to just like to sort of say, okay, does this make sense for the robustness of the comparisons? Do the ones that are most closely related to each other share the same patterns of filled in dots? They have that inventory of signature proteins in the genome. I think what gets even more exciting though, is to then ask, well, what do these proteins do? And so they start to look at some of the functions here. And in particular, they lift up membrane trafficking. So membrane trafficking, this is, a feature that's sort of a hallmark or signature of eukaryotic cells, right? So we have these ancient engulfment events, like of what became mitochondria, but there's also all this compartmentalization. So the nucleus, where the genome is housed, is separate from the cytoplasm. The endoplasmic reticulum, so all our secreted proteins get trafficked by the membranes, get moved around in the sort of contents within them so that they can be expelled from the cells through exocytosis or engulfed into cells by endocytosis, sort of these hallmarks of what eukaryotes do. And so some of the protein families are, again, this alphabet soup, or acronyms. So the escort pathway, the COP1 and COP2 protein coats that decorate some of the membranes to sort of encode the ability to move these things around. And you find these things in the archaea. And that, kind of from a traditional cell biology standpoint, was a little surprising. I think we have this, in a lot of circles, we still have this sort of false sense that bacteria or archaea are sort of primitive related to eukaryotes. We're all just as modern as each other. These are all being sampled today in real time to infer backwards. But what's interesting here is sort of the presence or absence of these and how that might speak to sort of like, how did some of the key features of becoming a eukaryote come online, and were those present or absent in sort of the ancestors here? And so why that matters is you're nestling eukaryotes or proposing that they sit in one part of the tree versus another. You're looking for that sort of ancestral relationship where these things existed, or where sort of the parts list was online potentially in the ancestors based on the fact that they're here today or still shared today. And this is a little bit of an echo, I would say, of Nicole King style science. So we've had Nicole on the TWiEVO podcast before. She studies these interesting critters, the choanoflagellates, and there makes a case that we're now much more modern times. We're in the eukaryotes in the sort of origins of multicellularity, when eukaryotes went from single cells, which is what's happening in that last common ancestor with the archaea, to then multicellular, like we are today. They're still single cell eukaryotes, but she uses the choanoflagellates because of their sort of phylogenetic relationship to then ask what are the sort of key components that they had on hand in those transitions, sort of massive transitions of life or cellular life moving forward. And so maybe an echo of that, asking about what are the membrane trafficking proteins that you find in archaea that are shared with eukaryotes. And in fact, that's kind of one of the surprises, or, this isn't new necessarily to this study, but it's becoming coming into more sharp focus, is that you actually have a lot of these sort of quote unquote sophisticated proteins involved in this cell biology. And I think the traditional view is that, actually those were donated from bacteria or co-opted somehow by the eukaryotes. And that's true in some cases, but the sort of archaeal diversity here, what it's telling us is that they have a lot of this parts list, or these tools, these protein machines, these components. And so it's actually very easy to explain that relationship between us, our ancestors in the archaea, through that sort of chain of custody of biology, where they've donated or these sort of collections of genes have come through that descent from that relationship of a last common ancestor.
And these are genes selected based on essential functions in the cell, right?
Exactly right. Yeah. And kind of hints at this, actually archaeal biology is studied so little. There's just, there's a few groups. I mentioned Rachel Whitaker at Champaign-Urbana in Illinois. And there's many groups, but compared to, how many, for example, cancer biology labs are, the number of archaeal biology labs it's a fraction of that. And so there's a lot of like core cell biology or just understanding how these things work that I think could be really valuable, sort of ironically enough, probably in understanding the cancer cell. There's some secrets hiding in our sort of distant cousins here, in a sense. And so they look at this a little bit too, not only through the lens of some of those, kind of cellular processes in terms of the cell biology here, but also asking about the presence and absence of some of these gene families as almost as characters to sort of make that case for the relationship between these major groups of archaea and the eukaryotes. And so here they're making some guesses actually about what they think the last common ancestor, like how it sort of operated as a cell based on the absence or presence of some of these sort of key features or that collection of genes that encode that. And so the guesses they make, one is that the last common ancestor of archaea and eukaryotes was a heterotrophic fermenter. And that's based on the fact that all of these archaea and eukaryotes are missing a major pathway in bacterial metabolism, the WLJ pathway, or the WLP pathway, Wood-Lungedahl pathway of bacterial metabolism. You don't find it in the archaea, you don't find it in eukaryotes. And so the idea was that as the last common ancestors of bacteria and archaea split from each other, it was lost in those early predecessors of the archaea of today. And then this is, so I think they do another kind of a similar treatment on carbon metabolism, a little bit with the electron transport chain. And that gets at sort of this alliance or that sort of tie between archaea and eukaryotes. And then finally, it kind of returns to this, well, where was this all happening in terms of the environment that these guys were in? And the notion is that the original archaea were in these very thermophilic, hot environments, these thermal vents, et cetera, but then radiated to more moderate temperatures, these mesophilic scenarios, where the Hedal archaea, the closest relatives to the eukaryotes that they propose here, with some of this analysis, that kind of, certainly like an intuitive sense, right, that probably our very ancient ancestors weren't living 3,000 meters under the sea at a black smoker, but instead were kind of like, hanging out more like at Yellowstone National Park or something like that. And so, so who knows, but really interesting, I think, to kind of speculate and tie some of these patterns together, both through the gene sequences and that sort of traditional molecular comparisons pioneered by Carl Woese and others, all the way to the the sort of idea that there's all these gene duplication events that are sort of potentially hinting at how the eukaryotes really sort of differentiated themselves from the rest of the archaea. They do a little bit of analysis there as well to say that among the modern archaea, some do a lot of gene duplications, the ones more closely related to us, and the ones that do less are sort of more distant in terms of the relationship. And so that could be another shared feature in a subset of archaea that sort of help to motivate or not motivate, but provoke basically the evolution of eukaryotes here as well.
So they're thinking that the gene duplication and less gene loss is somehow a requirement for becoming a eukaryote.
Yeah, and I think the duplication in particular. So that there, the idea might be that you can neo-functionalize that. Neo-functionalize. Yeah. You can bring up new functions by having sort of a backup copy to experiment as mutations and natural selection are acting. But
which we've talked about before on TWiEVO.
Yeah, exactly. Sort of one of the core. Yeah, exactly. Kind of a core mechanism for evolution to act. Yeah, exactly.
Yeah, it's interesting that they can infer the complexity of the archaea from this kind of analysis. As you said, we shouldn't think that they're simple. They have a lot of complex protein targeting pathways, membrane trafficking, genome maintenance, and so forth. So you can see the path to eukaryotes.
Yeah, exactly. And these are the success stories of like biological life. So these things have persisted for 2 billion plus years, right? Like cells still these are essential things sort of in the mechanics. I mean, that's also, I mean, I think it's kind of easy to look around at the diversity of life in very different environments and be impressed with how different everything is. But I think that, part of the story here too is the surprising like similarities that persist over long distances of time as well. It sort of makes it possible to even speculate about these origins, is the fact that we still share things 2 billion years on. That's a really wild amount of time for something to persist. Yeah.
How much sharing do we have with bacteria?
Well, so that's a whole other topic, and a really great question. That's kind of, I think in this paper in particular, in this treatment, is sort of pushed to the margins, is like, so horizontal transfer. So one of the tricks here is that if you're depending on everything that you share, you're sort of making a big assumption that it's all vertical inheritance, meaning that if the ancestor had this, then it was passed from generation to generation as sort of populations diverge. But there's, there's this whole other major mechanism of evolution that we're still grappling with how pervasive and important it is, which is horizontal gene transfer, which is you break the rules. Instead of inheriting something from an ancestor, you just either capture it or it gets donated or forced on a genome between species, this sort of horizontal. And so here you can have, certainly in the endosymbiotic event where two genomes are now like in the same cell, they're like very pushed together basically. But even just in open space, as organisms come together, microbial populations, like for example, like our guts, right? Here's like the microbiome, another sort of massively complex scenario. Or even in single cells, if ancient archaea or eukaryote is engulfing other archaea, other bacteria, viruses, etc., there's sort of this almost pathway, whatever you want to call it, of genetic communication where genes are being shuffled between genomes here, horizontal gene transfer, where there's no sort of descent, or like it's not mother to son or mother to daughter. It's just brother to sister and all across the way. And so that kind of any tree gets scrambled because now you have all of these sort of side shoots. And so that's over 2 billion years. That's also a big part of the story that was not ignored, but sort of pushed to the sides in this analysis.
And if you're wondering, yes, the archaea have viruses. They're called viruses now. They're not called bacteriophages. Some people make that mistake.
Yeah.
But don't, because you think somehow they're bacteria-like, but they're not. And they have viruses. Bacteria have bacteriophages, archaea, and eukaryotes have viruses.
That's right. Yeah.
And so it's interesting here, Nels, is I'm looking at the methods, and they sampled old. They used old samples basically that had been collected quite a while ago. So, for example, one set of samples is from the Tibet Plateau in Yunnan Province.
Yeah.
Interesting part of China that's in the news for origin of SARS-like coronaviruses in bats. 2016, they were collected. And
Yes.
so some of them were from 2002. These are all data sets that have existed or samples that have existed, and they're just taking advantage of it. It's really, really interesting.
I totally agree, and I'm glad you're bringing up that point because, we're kind of in this era where we're swimming in data. Like the sampling that we've done is actually still pretty modest in terms of all of the biology out there, but but we're as scientists, we're such a small community relative to the sort of diversity of life out there that there's and so you might have all these collections that also you're kind of waiting for the technology to be right as well. So something collected 10 years ago, let's say,
Yeah.
even if you analyzed it then, analyzing it today is like there have been three revolutions or something in the ability to squeeze sequences out of really minute amounts of nucleic acids and to sort of cover the entire thing, and then obviously the computational power to crunch through these massive and complicated data sets. A lot of software developed to basically put this together, and so these old data sets are like kind of you can breathe new life into them with the analysis that's possible today.
In many cases, they got samples. So, for example, here is a sample from the South Hydrate Ridge that was collected in 2002, 12.4 and 14.96 meters below the seafloor.
Yeah.
And they got 10 grams of each sample. So the people who had collected them were sharing them, and they extracted the DNA and did the DNA sequence analysis in this study. So this is all about sharing as well, right? You give it to people who have an interest and who have the ability to look at it in a way that is going to give them insight, as we've talked about here.
Yeah, and I think your second point on the ability to do that. So with these rare samples that are really hard and expensive to collect, you don't want to sort of turn over that 10 grams and end up with like basically 100 base pairs of just junky sequence. You really want to maximize the information that you get out of there with robust techniques. And this group has proven that they can really do that over the last decade plus. And so I think that's, exactly. That sort of helps to facilitate the collaborations, the sharing, and the ability to squeeze really good info out of this work.
So I mean, clearly this is an ongoing project, right? So as people get more samples and do more of this kind of analysis, what will happen? I mean, right now we know that the eukaryotes are nestled in there. That's clear, right? That's not going to change. It will just get more detailed.
Well, so that's a good point, I think it's a great open question. And so sort of on two sides. So on the one side, you could say, okay, you've added 60 more species. You've done sort of, some new analysis, use some new tools that way. But so has that really kind of broken open the story in a new direction? And of course, the authors are claiming it has. I think I agree. I mean, it caught my attention. Whether it's like, I don't think it's not sort of the same scale of a breakthrough that the original discovery of the Lochia archaea was, right? That opened up like a whole new sort of set of life that we'd never glimpsed at before. Here, the metagenomics, as you're saying, it's just sort of filling in the branches, or in some cases adding a new sort of whatever arbor of branches onto the tree. But so that's the open question. Is there another sort of breakthrough event where we sample a place in the world that we haven't looked before, or a species hasn't, maybe we have already sampled, or maybe there's, maybe that sample was collected in 2016, but we haven't gotten to it yet. And there could be an entire new form of life hiding in there, that sort of, and so that's the thing is every time even as you add more diversity through the metagenomics approaches, using the comparisons of what you already know, that can change some of the, how the branches arrange with each other. If you add an entire new thing in there, that could like kind of radically redefine the relationship. So I wouldn't put it out of range that sort of nestling relationship with that set of archaea, that could be sort of blown out of the water in the future. as well. So, but it's a good question. I think the probability that it's blown out of the water is less likely the farther that we get.
Yeah.
And so we might be pretty close to the truth.
So I was at ASM on Sunday. I heard Andy Weir interviewed by Kate Rubins.
Mm-hmm.
So Andy Weir, of course, is the author of The Martian and Hail Mary and other books, right, about space.
Yeah.
And Kate Rubins is an astronaut. And they're talking about finding life on other planets. You know, there's some rumblings that we might go to the moon, that we might go to Mars eventually. And she said that we want to look for life. And he said, there's no life there. It's all burnt. There's nothing. You know? So I'm thinking, why would you want to look for life? But obviously, you want to sequence the genome and see how it compares to this. Is it in any way related, or is it totally different, right? If you could find it.
Agreed. Or different is it based on a different chemistry? Do we converge sort of on replicative life forms with totally different sort of nuts and bolts there? No, I think it's like, actually in two ways. So I thought your story might go the other direction, which is not like, no, there's no Martian life, but, that we need the sort of creativity, the imagination of science fiction writers to sort of open up our own brains, to like allow the space for like radical new things to emerge as scientists as curious people. It just sort of feeds that I love how the creativity of like what you can even imagine like in real life. I think if you go far enough in the universe or over enough time, we're probably even only capturing like, our wildest imagination probably only captures like a small fraction of what exists.
What was interesting is that she wanted to explore. Of course, that's the astronaut view, right? She wanted to go look for life. And he said, look, here's my view. On Earth, wherever you look, you get soil, you get dirt, you get water. Anywhere you go, you see life. So he says, on a planet, if there's life, it's going to be everywhere. And we know it's not everywhere on the moon. So it's not everywhere on Mars because we've sampled all over. There may be something, but on the moon, it's probably just blasted away by irradiation.
Yeah,
it's maybe the same for Mars. So he was very pessimistic. But you could get samples and look, I guess, for remnants of DNA. Although I don't know if it's going to last. Who knows?
Oh yeah, no, who knows? Yeah, and I agree. So like in that sort of very small sort of sampling of some planetary bodies or moons, et cetera, around us, fair enough. But I think, again, it's sort of that, like how wide do you cast a net, and like considering sort of the tip of the iceberg or even the tip of the iceberg kind of scenario. I was reading recently, I don't follow this too closely, but there's a moon, I think off of Saturn, that has some features that were being lifted up as potentially being amenable to life. It's not potentially blasted out by irradiation, et cetera. But but
the problem is that we can't go very far. It just takes too long.
Yeah.
But here on Earth, we can sample a lot. It's within our ability to sample. It's just a matter of money and people and so forth. So it is more attainable than finding out if there's life somewhere else.
Yeah. I mean, I definitely agree. Yeah, agreed. I mean, I definitely agree with the notion that like there's plenty to explore here as well, or to protect here as well, versus thinking about, oh, well, maybe we just set up some colonies on the moon or Mars and then just hang out and everything's fine. Like, there's one Earth, and one that's sort of the way forward for our species, for instance. Yeah.
But anyway, it's not clear that we are going to the moon because she said they haven't put money aside yet, and it's expensive and it's dangerous. It's not straightforward.
No. Yep.
I mean, she was saying that, they get trained for medical emergencies. She said, but there's some stuff you got to ship someone back to Earth, but that takes 5 days from the moon, and forget about it from Mars. You're going to have to learn how to do it there. Anyway, it's all very interesting. Let's see. We had some good questions here.
Oh yeah, let's pull up a few.
Here, Trevor says, "Is it possible that DNA arrived from space?"
Oh, now we're talking. Yeah, so this connects our first conversation to the second one here. Yeah, what is that? There's a term for that, zoospermia or something like that.
Panspermia.
Yeah. I don't know. I haven't followed that one too closely, but I wouldn't rule it out. like the same way, like there's all this horizontal gene transfer, could there be galactic gene transfer? Certainly like the prevalence of that. I mean, maybe that gets back to the author's, what was the name of the author that you mentioned?
Andy Weir.
Andy Weir. I mean, that gets back to his point, sort of like, give it the sort of is it in front of your nose test as a first take. But yeah, no, I think that's an interesting active area of research where DNA is coming from, or other sort of chemistries of life that could be sort of like right under our noses, or part of like a meteor rock or something like that that gets through the atmosphere.
I don't know. I mean, it's DNA going through space. There's a lot of radiation. It's going to trash it, right?
Yeah.
I mean, that's how we make mutants. We can irradiate cells, and so I think that's a long shot. I like the idea that it originated here, right in front of us, right? It's
Yeah,
quite
for
clear,
sure.
but you can't rule it out. Yeah. Here is another one here. Marianne says, if archaea are extremophiles, could they be more similar to extraterrestrial life if there
I
is?
love it. Yeah. Good. Yeah. So that's another, it's a great question. I think like there's a lot of research out of NASA that's sort of like where astronomy and biology sort of meet. That's an interest, like really, and speaks to some of these origins of life and extraterrestrial life. There's some good reasons to mingle those ideas, or to sort of gain scientific progress that way. And so, thinking about how things look on other planets sort of makes sense to frame, in some cases, how did life originate here? Those like living obviously in extreme environments, just as we've been discussing, and what can make it. There's a lot of potential, as the archaea are teaching us. If you can live at 3,000 meters under like these ridiculous conditions, a lot is possible, and stuff that we would have assumed wouldn't be possible, right? Even in the last 2 decades or something like that. And so I think it's really wise to be open-minded about the possibilities, and that we're sort of still have a pretty limited view about, what you can sort of weather in terms of, and sort of continue to replicate.
So Pete says, DNA replication enzymes, are they core functions? Yeah, they are.
Yeah, those are in there. That's part of it. So I think there's one of the supplemental figures, and worth it, by the way. So if you can, put up with a sort of alphabet soup of like acronym after acronym, there's one of the supplemental tables lists the 57 genes, excuse me, encoding the proteins that they put into their, their comparisons to build those trees. And you can see the whole kind of parts list, and it crosses over a couple of those core functions, including replication, some of the polymerases.
All right, Jeff says, dumb question. No dumb questions, but does the advent of immensely complex immune systems prevent a huge, broad branching evolutionary event like the mitochondrial symbiosis from happening again?
That's a great question. I don't think so, but I think, so ultra rare events, I think we also, we should be a little bit cautious. Like when what do we mean when we say immensely complex immune system? I mean, currently you can open up the pages of some of these magazines, like right next to this article is probably in that same issue, might be something about a bacterial immune system that someone just, described or discovered. And that also, I think it's a nice illustration of, so microbial life, generally single cells here. But that doesn't mean primitive life. The immune systems, or the sort of the biological sophistication, is modern. They've had just as long to sort of evolve as we have. Yeah. And so, redefining kind of complexity, intelligence, sort of all of these features into, So I think the answer is probably yes. So that, there's always this balance in whatever the complexity of the immune system to, that gets sort of litigated in just the replication, both of the, the collision between two genomes, the thing that's infecting the host, or, even how you define that, can be a little bit slippery. But if your immune system is too, regardless of the complexity, if it's too intense, the autoimmunity that will happen will start to drag on that population. And then, right. And so there's some balance there between not killing yourself, but recognizing something foreign and sort of getting rid of it if it's weighing on your own replicative success. And so somewhere in that balance, I think, is just that possibility of these rare events happening where a symbiosis might sort of take up in that. But it's a great question. And honestly, like what that balance is, or what that means for the tempo of symbiotic events, I think is really interesting.
Yeah.
I think probably some of the most exciting work here is in insects, where modern symbiotic events are happening sort of all of the time between, for example, insects and bacteria, like Wolbachia and Drosophila and other systems where we have all these experimental tools.
Andrew says, I love this synchronicity. Eukaryote means good seeds, nuts, and they're making a tree from its evolution.
Yeah,
good.
Some good poetry there. I agree.
Lori writes, I'm 2/3 through the Tangled Tree, wondering what you think Carl Woese would make of the paper. Yeah, we mentioned that earlier. I think he'd love it because he was a data-driven person, and he made conclusions based on the data. He would never say, no, I believe 3 domains, and therefore nothing else could ever disprove it. That some people are like that, for sure.
Yeah, good point.
But Carl was not, and he would see these data and go, yep, well, we already know there are 2 domains, as Nels said, but this reinforces it, and I think Carl would be all for it, for sure.
Yeah.
What do you think, Nels? Yeah,
100% agree. I think, and if anything, it's sort of a tribute to some of the techniques that he and others of his time were pioneering. That this is like, it still builds off of that. It's just as new data comes, and that's what we're describing here. What you're describing, Vincent, is the scientific process. That's the whole point, is like you want new data to come in to revise what you put up before. And it's not that science has always operated that way. In some corners, it's not operating like that today. It's sort of the false sense of authority. This was your colleague, Stu Firestein, Failure, Why Science is So Successful, and Ignorance, I think is this other book, where, some of the stories that he tells about or recounts of the history of some of like medical science, where it was some dude who just said, like, here's how the blood circulates through the body, and it was wrong, but it was so like kind of ingrained into the authority of that one person that usually there's these sort of, kind of tongue-in-cheek ideas that science moves forward when when an authority figure dies. In this case, it was like generations of people kept that false view sort of in this like, memorial to that person or whatever. And so that's not science. That's sort of humans being weird. Whereas when new data comes in and you refine or revise or totally like blows your sort of original idea out of the water, that's like best case scenario, actually, because now science is moving forward in an interesting way.
Indeed. All right, Marianne asks, do we have any of the same viruses that the eukaryotes had? Well, I don't know. Maybe you're getting confused with bacteria and archaea and eukaryotes. So we are eukaryotes, right?
Yeah.
And presumably some of those were in the earliest eukaryotes, but who knows? It's a long time ago, right?
Yeah. If I had to make a prediction here, I would say yes. I think a lot of this, and maybe the best modern examples I can think of, are some of the giant viruses that have emerged in the last about the same timeline, maybe even just a few years before all of this diversity of the archaea. But, so some of these viruses can infect both, or have the gene. If you look at the genomes of some of these giant viruses, they're chock full of genes from bacteria, other eukaryotes, and archaea. So they've sort of have been sampling from all 3, or sorry, I almost said all 3 branches of life, all 2 domains of life by this definition, all the big categories here. And so, that either implies sort of the ability to infect broadly, which has been demonstrated in some cases, or, certainly like these very intense sort of collisions between all of these species at the same time. And so anytime like genomes get together, the chance for cheating and for exchange of genetic information is almost assured. And so I think, although, it's a great question, because it's also not like, I don't want to sound like, oh yeah, of course that happens. There's also specialization, but especially as you go. So certainly there are viruses that can infect a broad diversity of eukaryotes, from fish to humans, for example, in sort of a history of that. So fish get influenza virus, for example, very different strains, but the same virus classes. Like the viruses of bacteria are in general very different. The phages, which is why, phage therapy is being proposed as potentially a novel or a different antibiotic, is that generally those are not going to infect us. And so it's a pretty safe intervention. And so there are barriers here as well, in species
Yep.
specificity
Yep.
of viruses that also is at play. Yeah, great
I
question.
like that when genomes get together. So that gives me an idea for the name of a bar, the Genome Bar, right?
There you go. That's right.
Oh, not that I'm going to do that in my lifetime. I got enough with microbe TV, but somebody make it called the Genome Bar. Would you go to a Genome Bar, Nels? I would.
Yeah. How about the genome bar at the incubator? I'd come right over. We could.
Yeah, you bet.
Yeah.
Andrew says we should be looking on Earth for other types of life as it is the most likely to be in an environment where life has emerged. Sure, absolutely. I agree with that. And then that brings us to Marianne, who says, "Is it kind of strange to think that we're the only existing life? It's just too big." Some people, it depends who you talk to. So I think life was just a crapshoot, but some people say, well, there's so many potential crapshoots out there that could be other life. But I am secure in saying no because we'll never know. It's too far away.
Yeah, I don't know. I'm a little more open-minded, maybe somehow. So I think like because the universe is vast, I just don't like for me at least. I don't think my brain is good at like 100 years of time, maybe in terms of like what does that mean or comprehending that. Maybe reading history certainly more than that, but not a million years. Like my brain, I don't think can wrap its whatever synapses around that. And same for space, right? So like our own solar system and sort of growing up with learning about Neptune, Saturn, Pluto, whatever. But then, like even with some of the new telescope and some of those images or the distances that they're talking or the light years that they're talking about, again, I feel like my brain sort of falls short in terms of like comprehending all that's possible sort of in time and space. And so that's why I would lean towards yes, it's out there. But yeah, whether we get there or the time frame that it takes for those connections to happen, I think that's a very fair
that's the Fermi
question.
paradox, right?
Yeah, exactly.
Never know. Can't prove or disprove. I mean, I don't hide in that. I just think when I read about how we think life emerged, man, it's such a crapshoot that it happened. And so, but maybe if you got enough numbers, it overcomes that. I understand that.
Yeah.
Pete says, is other life opposite chirality, left versus right-handed amino acids?
I love it. Yeah. So there's one of my colleagues here at Utah, a biochemist called Michael K. He makes D peptides. So there's the chirality here. There's L and D. And so he's actually, one of his sort of dream projects is to make D. coli, where all of the amino acids are the mirror image in all of the proteins. So a life form that would be a mirror image. He actually uses that concept of chirality for a more modest sort of intervention, which is to make peptide therapies for things like antivirals that don't get chopped up by proteases. They have the same biological properties, but they're not recognized by proteases because of that mirror image. And so he's had some success there, actually, and pretty interesting question. Interesting. So yeah, and that's another kind of one of those like coin flip questions. Let's say there is another life form that our species comes across within the next 100 years or something like that, and in assuming that it's based off of amino acids, then that would be, I guess, under some principles, it'd be a 50-50 coin flip, whether that would just be the mirror image, basically, at the molecular level.
Tom says, whatever we find will correspond to fundamental chemistry and physics, and that's quite well understood. Be creative, yes, but not preposterous. I would agree with that, right?
Yeah, right? I agree. There's a great line from Lee Van Valen, who is an evolutionary biologist at the University of Chicago, and who proposed some of the Red Queen, actually coined the Red Queen hypothesis, or part of it. And he has a great line. I might kind of not get it exactly right, but it's like, keep an open mind, but not so open that you'll like spill over into the sort of preposterous space. And that's a balance too, right? So to do kind of really kind of creative work, but to also sort of, constrain yourself to what's possible. Yeah. Great point. Can be a tricky balancing act. If anything, I'm not worried about that. Like when I think about NIH funded science, if anything, we're kind of on the other end of the spectrum where can become like, echo chambers of fields within fields. And sort of, there's plenty of room for creativity to add creativity to the mix in modern biomedical science, at least.
Pete says amino acids certainly could have come from space. They may have seeded chirality of life on Earth. Yeah,
Some
can
cool
amino
ideas out
acid
there.
make
Yeah.
it? Would amino acids make it through space, Nels?
Depends on what they're traveling in.
I suppose. Yeah, maybe in the center of a rock would protect them, right?
Yeah,
I don't know,
exactly.
man. It's a lot of ifs. I like
A
to
lot of
have simple assumptions, you know.
It's a lot of ifs, but it's also a lot of space and time. And
Okay.
yep, yeah,
Oh, Marianne would go to the genome bar.
fantastic. Well, let's meet up.
Well, maybe I should make a little genome bar here within the incubator. Then when people come, we can have a drink at the genome bar. Rob says the JW Space Telescope can see the atmosphere of distant planets.
Yeah, and so this opens up the possibility, at least sort of kind of the next sort of scale of exploration, I would say. I agree with this, where, so if you see atmospheres, right, so that starts to get at some of the Earth-like properties or the protective, nature that you need from the harsh sort of environments of space. And so that's, exactly a core. And being able to like detect that for the first time at that distance really opens up the possibilities of starting to get a glimpse at where some of these rare events might be unfolding.
If the universe is infinite, then there must be a case where Nels is in the incubator and Vincent is in Salt Lake City.
The chirality of TWiEVO somehow. Yeah, that's right. The mirror image.
That makes sense. And Sam says it could be proven with an example. If we find life on Mars with no DNA, that would do it. What do you mean, like microbial life, something that's growing, without DNA?
Oh, yeah. No, I like this point. So, that's the thing about all of these conversations is we're like, oh, yeah, it's not, we don't think it exists or it's not possible or it's sort of until it does. And so, and even like if we return to just today's paper, like 10 years ago, Loki archaea wasn't described. It was hiding out, it was living happily at these thermal events, but we had no idea about that. But then once it emerges, then that's the other thing about the human brain, I think, is that once we learn something new, it becomes almost boring, like in a very short amount of time, which is like a very interesting, probably adaptive trait, but we're so fast at adapting to new info and sort of like taking it. And so, but maybe the flip of that coin or the other side of that coin is that, something is impossible until it exists. And then it sort of becomes almost second nature. And so, kind of weighing that into the situation as well. I mean, like whatever, 2 years from now, maybe we'll be super comfortable that there's like this sort of alien life form that now that we just like intermingle with at the genome bar or something like that. Who knows what'll happen?
Let's see. Could you make different DNA with different bases in the ladder?
Oh yeah. So there's like, this is happening. So, right. So there's also, I think, some really fun work in, I don't know if I might get the terminology a little bit wrong here, but like unnatural amino acids, incorporating that into chains. All kinds of messing. I think probably it's easier to mess around with the R groups of amino acids and still build like peptides and proteins. But you can mess around with these like chemistry or biochemistry. There's some really great labs that have kind of in the space of synthetic biology, I would say, who are playing around with this and messing with the genetic code, et cetera.
Another vote for the genome bar. Yeah, I had a good idea. Genome bar, where when genomes get together, in a bar, right? Where else?
That's right.
The Space Telescope can see exoplanet atmospheres just in the nearest star systems. Okay.
Yeah.
So that tells you what they're composed of, I guess, Nels, right?
Correct. And that they're, might not be as rare as maybe we thought. I think that's where that technology is sort of pushing human knowledge. Yeah.
I don't mean to not be adventurous, but I just am amazed by the randomness of how life emerged here. That's what I'm based on. But for sure, go explore. You just can't go very far with humans. You need to put robots. But then, a million years later, it's going to... It'll tell us what's going on, right? That's a tough one. All right, Nels, you want to do some picks?
Oh, yeah. Let's move into science picks. So mine for this episode is we're also here. So it's midsummer. It's also national holiday 2 days ago on Monday, Juneteenth. And the journal Cell it's not a full issue on Juneteenth, but it had a collection of essays from brown and black scientists on the occasion of Juneteenth, now a national holiday. And one of the featured articles was Juneteenth in STEM and the barriers to equitable science. And so there's 50 plus authors here. I think A.J. Hinton, who's an assistant professor at Vanderbilt, sort of helped organize this group. And I think a really important and timely essay. I pulled out just a couple snippets here from the article just to give a sense of it. And again, open access, take a look. Very much worth digging into. So in addition to cultural erasure, undervalue, isolation, stereotype, threat, and tokenism, Black scientists face many obstacles to attaining an education and persisting in the fields of science, technology, engineering, mathematics, and medicine. As the official correspondence to the White House states, Juneteenth not only commemorates the past, this is the abolition of slavery, it also calls us to action today. Juneteenth is a rallying call for all, but it is especially a call for action from scientists. Even though scientific innovation prospers from a richly diverse field, science has historically existed as a bastion for harboring racism. And so, this is, I think, both, calling out some of the historic and also persisting challenges. I think the field of evolution in particular, as we've talked about, Vincent, we had Joe Graves on a while ago, and I've talked about in some other episodes, like the underrepresentation in evolutionary sciences is, I think, especially striking. And so not only calling attention to it, but taking action, I think, is really, and getting creative and innovative in doing that is really important to fostering and supporting science ahead. Cool. So, that's my pick of the week. How about you, Vincent? What's your science pick of the week?
So I have a website for the Tara Oceans Explorer. So Tara Oceans is part of a larger exploration thing called Tara Ocean Expeditions, and they have a bunch of expeditions all over the world. Tara Oceans, basically a boat sailed all over the place and collected many samples. It's been doing it for 4 years. The Tara Oceans is a 4-year-old version. And it's a boat called the Tara Oceans, and the boat, it's a schooner actually. It went to sample all the seas on the planet to study plankton, and so it's the organisms that drift in the ocean. Plankton is kind of a catch-all phrase, right? Anything that's drifting in the ocean.
Yeah.
Microscopic, that is, not a whale that's dead and drifting. It's not plankton. It has to be microscopic. So they took 35,000 samples of viruses, algae, and plankton. And, of course, they do genome sequencing, and they look to see what it is and so forth. And this was published in Science in 2015 in 5 articles. But it's all the data are accessible to people, and you can take a look at it. Let me just show you what this looks like here. This is the website.
Cool. Wow.
Tara Oceans, an expedition to the heart of biodiversity in the world of plankton. And so they have a nice summary of all of the steps, and they have the team here, right? The director of the expedition, and so forth. And there's also there's a map here of the here it is. There's the it went all over all the oceans were sampled. So it's very cool.
Yeah, cool. Agreed.
Very cool expedition.
Yeah, and they say here nearly 35,000 samples containing viruses, algae, plankton collected and analyzed. And an echo of our paper today, right? Where that's where a lot of these archaeal species are coming out of deep oceans, and in a lot of cases, and very neat.
It's a cool thing to check a look at and explore. I just want to highlight a couple more comments here. Andrew says, xenobiology, the branch of science with no evidence yet.
Exactly.
That's very good. That's very good. And Marianne, who just left, I truly appreciate you professors and the rest of the community here. It's a wonderful community that we've built here with our live streams.
Thank you.
Yeah,
glad to have you here.
And Pete says, I think there's just one black chemistry prof in the UK. It's terrible, right? Very bad.
It's so out of balance. That's right.
We need to fix this.
Yep.
All right. I believe that's it for this episode of TWiEVO. We don't have any emails this week, so we answered your mails, so that was really cool.
Yeah.
That is TWiEVO. Microbe.tv/TWiEVO is where you can find the show notes. If you want to send us questions or comments, TWiEVO@microbe.tv, or you can just come here. We'll be back in about a month.
Yep.
If you like what we do, consider supporting us. We are a nonprofit organization. We exist to communicate science, which I think is really important these days because people think science is a debate. It's not a debate. You get results and you make a conclusion. It's not a debate usually. Of course, it can change if you get different results, but it's not a debate. Not going to debate the origin of SARS-CoV-2 because we know what the origin is. So if you want to support that kind of science, microbe.tv/contribute. Nels LD is at cellvolution.org on the Twitter. He's at L Earlybird. Thanks, Nels.
Yeah, thank you, Vincent. Great to be together as usual. And we're just starting to scheme on TWiEVO 100 coming up by our calculations in March. So stay tuned. I think there's some fun stuff happening as we're starting to plan. Thanks, Vincent. Great to be together, and see you next month.
I would like to thank everyone for coming today here to listen and to learn. And I want to thank our moderators. By the way, Les showed up here quite a while ago, but I didn't get to mention him. Thank you for moderating. And he reminds us tonight will be office hours, 8 p.m. Eastern. That is where I chat with you guys, answer questions. We should get you on office hours one night.
Oh, yeah, I'd love to come.
Maybe the next one we'll get you on. And we'll try and do it a day we don't do this one, so you don't have to do two in a day like I do. But folks would love to have you on office hours. It's a nice chat. 8 p.m. Eastern. I also want to thank Barb Mack for moderating, Andrew for moderating, Tom, who has left, Les, as I said. Steph was here originally. She might have left. Thank you all for moderating. We really appreciate it.
Yeah, thanks all.
Music you hear on TWiEVO is by Trampled by Turtles. Let's get that music going here.
Fire
you can
it up.
find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, a podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious. Bye-bye.